So motor system, the overview, lower motor neurons in brainstem and spinal cord, upper motor neurons in primary motor cortex and other cortical regions, motor actions planned and controlled by association cortex, basal ganglia, and cerebellum. So motor actions planned and controlled by association cortex, basal ganglia, and cerebellum. Motor system pathways and brain dissection video. So lower motor neurons and the motor unit. Lower motor neurons in brainstem and spinal cord are arranged in groups according to individual muscles. Their axons leave at the ventral roots of the spinal cord and go to the neuromuscular junction of the individual muscle fiber. This is called the motor unit. Lower motor neurons are arranged systematically. There is systematical mapping that is the same as anatomical mapping. For example, in spinal cord, neurons for axial muscles like posture and torso are medial, and distal muscles are more lateral. So neurons for flexors are dorsal, and those for extensors are ventral. So flexors, dorsal, extensors, ventral. Axial distal mapping is reflexive of the location of these pathways that descend the brainstem and spinal cord. There are three kinds of muscle fibers and motor units: red and white muscles. She says dark meat, light meat, ew. Motor units are slow twitch, fast twitch, fatigue resistant, or fast twitch fatigable. So we think, stand, walk, jump. Motor units are recruited in order of size. So we have the size principle. It grades the force of muscle contraction, recruits small to large. There's slow twitch to fast twitch. So small, slow twitch, large, fast twitch. Why our muscle movements are so smooth? So upper motor neurons are control systems. Upper motor neurons control lower motor neurons directly and indirectly at the same time. Basal ganglia and cerebellum control motor via thalamus by influencing output from cortex to brainstem and spinal cord. The basal ganglia and cerebellum also project to brainstem nuclei. The association cortex, basal ganglia, cerebellum control choice, design, and monitoring of motor movement, but do not have a direct influence on lower motor neurons, which are indirect. So lower motor neurons also affected by reflexes and motor programs. These are automatically putting one foot in front of the other when you walk. So motor programs, sort of like reflexive walking. Motor tracks, descending motor pathways recap. Vestibulospinal tract, or postural adjustment and head movements. Corticospinal tract, or voluntary movements. Reticulospinal tract and rubrospinal tract, which are alternate path for voluntary movements. There's also the tectospinal tract from superior colliculus to cervical spinal cord for reflexive turning of head in response to visual info. Corticospinal and rubrospinal tracts end in lateral spinal cord and influence distal muscles. All others end in medial spinal cord and influence axial muscles. So modulation of the motor cortex. This is an oversimplification. Association cortex decides that a motor movement is going to happen. Premotor cortex devises a plan for the movement and passes the plan on to the primary motor cortex. M1 then issues the command via the brainstem nuclei and interneurons to lower motor neurons to fire. The premotor also sends some direct commands to lower motor neurons. Basal ganglia, thalamus, and cerebellum do not. They plan and monitor movement. So premotor can send some direct commands to lower motor neurons, but basal ganglia, thalamus, and cerebellum do not. They just plan and monitor the movement. So here we have the primary motor cortex, M1. So C is meta-analysis of fMRI studies asking subjects to use or imagine using or engage in cognitive tasks that might use these parts, and activation is mapped along the motor cortex. So the M1 cortex is somatotopically mapped like a homunculus. Areas that generate fine movements are given more cortical space than those with only gross movements. So you can see foot, eyes, arm, hand, fingers, mouth. We have Betz cells, named after Vladimir Betz. These are primary motor cortex that contain very large neurons. These are Betz cells. These are pyramidal neurons. The axons project through pyramids in brainstem to the spinal cord. And strikingly, only 3% of CST is axons from Betz cells. Only half of CST originate in M1. So axons project through pyramids and brainstem to spinal cord, but only 3% of the corticospinal tract is axons from Betz cells. So here we have the corticospinal tract. Note how much of the mouth region of the premotor corresponds to the Broca's. Primary motor cortex on precentral gyrus and deep within central sulcus. Premotor cortex anterior to M1 on the lateral surface has 4 subdivisions so far, and also has a somatotopic map. So there's the mouth, corresponds to Broca's area. Then you have the supplemental motor cortex anterior to M1 on medial surface. There are 5 to 6 subregions somatotopically organized. Both also project to M1 as well as down the CST through PLIC, M1 most posterior, then premotor, then supplemental most anterior to the PLIC. Mirror neurons are in premotor neuron area. So here we have a newborn monkey imitating human movements. So the newborn monkey is imitating human movements. Mirror neurons in premotor area fire both when watching the action and when performing the action, the premotor. So these mirror neurons in premotor ventral rostral area both fire when watching the action left and when performing the action. So DTI fiber tracking of CST, so cortical spinal tract, Axons leave the M1 and some cross CC to contralateral hemisphere. So they cross the corpus callosum to contralateral hemisphere, but most descend to corticospinal tract. So from M1 heads through centrum semiovale, as we see in B through D, and then through corona radiata in E, and then through the PLIC, or F, and cerebral peduncle and pons in H. DT tractography to show somatotopic mapping of CST. Fibers keep their somatotopic mapping from M1 through thalamus, internal capsule to brainstem. For example, the foot travels medially and the hand laterally. From motor cortex to hip, trunk, central sulcus, arm, hand, face, lips, lateral fissure. fMRI study to show role of SMA, PM, parietal association cortex in motor planning. S is given 2 cues in random order. Which target to reach for, which hand to use. Goal was to see which areas are involved in aspects of motor planning. So the non-primary motor activate during the planning and imagining of the movement, but the primary motor cortex activates most robustly during the actual reach and not really before. So regions responded to both cues. The dorsal portion of the premodal parietal association cortex, or IPS, and the supplemental motor cortex. In contrast, M1 only activates later after the planning regions have activated during the actual reaching. So you have the cue and another cue. So you have which target to reach for, which hand to use, and then the actual reach. Then we have the corticobulbar tract. So cranial nerve motor nuclei on brainstem also have upper motor neurons. The collection of fibers that leave the cortex and end in the brainstem through the IC and reticular formation are called corticobulbar tract. The bulb is an old term for medulla. This is especially true for fine motor control regions of the face, like the lip and tongue. This ends in the pyramids, and so this is the other pyramidal tract. The CST is for the body, the corticobulbar is for the head and neck. So CST, body, corticospinal tract, and corticobulbar is for the head and neck. Maybe think bulbar like bobblehead. So bulbar medulla for face and fine motor functions.